This is a production of Cornell University.
And thank you guys all for being here. I am really excited to share with you what I've been working on for the last 6 years in Polerovirus research. Oh, and I just realized my mic's not on. Okay, better. Okay, so my research focuses on plant pathogenic viruses in the genus Polerovirus and the family Luteoviridae. Poleroviruses have a single-stranded positive sense RNA genome, meaning that the genome itself is protein coding. These viruses move in both aphids and plants as icosahedral virions. Unlike many other plant pathogenic viruses, Poleroviruses are transmitted exclusively by aphids in a circulative, non-propagative manner. Aphids deposit virions directly into the phloem, shown here in red, while they're feeding. The virus then replicates and moves systemically, but never crosses out of the phloem. This phloem restriction is believed to be an adaptation to facilitate acquisition by sap-feeding aphids. The symptoms caused by Polerovirus infection vary by host and species, but often include intraveinal chlorosis, as you can see in this infected hairy nightshade plant. Poleroviruses are an emerging problem worldwide. Shown here is a summary of the geographic distribution of new and emerging Poleroviruses in 2018 alone. Purple stars with pop-out boxes represent new Polerovirus species. The same year, Polerovirus species were reported for the first time on 8 new hosts, shown by blue stars, and in 7 new countries, shown by orange stars. Like other RNA viruses, Poleroviruses mutate and evolve extremely rapidly. Therefore, even species that have not been an issue in an area for many years can experience a resurgence if new strains emerge. Additionally, the use of insecticides to control aphid vectors is neither cost-efficient nor effective. By the time a grower realizes they have a Polerovirus in their field, it's often too late to contain the outbreak. Additionally, insecticide resistance is a growing issue in aphids. Fortunately, we have several well-studied Polerovirus species that we can use as models. Potato leaf roll virus, or PLRV, affects solanaceous plants and causes a net necrosis disease of potato that was once an enormous problem in the United States. Turnip yellows virus, or TUIV, is a pathogen of brassicas and is currently of economic importance on canola. Decades of research have generated an excellent toolkit for studying these 2 viruses, including infectious clones, which allow us to inoculate plants by agrobacterium infiltration, virion purification protocols, a wealth of homemade and commercially available antibodies, which are useful for ELISA, Western blot, and other applications, and protocols for rearing aphid vectors. Finally, I've labeled this box Virus Mutant Library, but the real resource here is Stuart Gray, who is one of the foremost Polerovirus biologists in the world, and we're fortunate to have him located here at Cornell, just across the street from our lab. Stuart has been incredibly generous with his expertise, his equipment, and of course his mutant library. Oh, I shouldn't do that. And this project would not have been possible without him. Another advantage to working with Poleroviruses in general is the small size of their genome, which is about 5.8 kilobases and encodes only 9 known proteins, which I'll walk you through briefly. P0 functions in suppressing host silencing, which is the primary plant defense against viruses. P1 and P2 encode a polyprotein and a fusion protein, which are important for replication. P3A and P4 function in movement in plants. P3 and P5 are structural proteins which form the capsid. I'll talk about these a little more in the next slide. And the functions of P6 and P7 remain mysterious. As mentioned previously, Poleroviruses have icosahedral virions. The capsid is made primarily of coat protein, or CP, shown in blue and encoded by ORF3. However, sporadic read-through of an amber stop codon causes translation through ORF5, producing a C-terminal extension called the read-through domain, or RTD, shown here in orange. The resulting read-through protein, or RTP, is incorporated into the capsid at a low frequency, with the RTD projecting outward from the capsid surface. The RTD is also found in an unincorporated form, which has been shown to have its own functions. Mutational analyses, primarily from Stuart's lab, have pinpointed regions in the CP and RTD which are important for virion assembly, aphid transmission, movement in plants, and restriction to phloem tissue. In spite of the many tools available for Polerovirus research, the structural biology of these viruses remains difficult to study. For starters, Poleroviruses are phloem limited, which means that they're located in tissue that's hard to lyse and hard to separate from other tissues. This also means that the overall virus titer in the plant is quite low, as most of the plant cells are actually not infected. Consequently, it's difficult to obtain a large quantity of pure virions. Because of these and other challenges, there is no publicly available crystal structure for any Polerovirus virion or structural protein, which limits our ability to infer how mutations impact structure. One technique commonly used for studying Polerovirus structural biology is an RNase protection assay. In this assay, virions extracted from plants are subjected to a short treatment with RNAse, which digests any RNA that is not protected by a capsid. After RNase treatment, the protected RNA is extracted and used for cDNA synthesis, and the presence of the viral genome is assessed by conventional PCR. In virion preparations that have not been subjected to RNase treatment, the genome is intact for both the wild-type virus and this delta CP mutant, which is a mutant that does not produce virions. However, the absence of the capsid in the delta CP mutant leaves the genome vulnerable to RNase, resulting in near total degradation of viral RNA after RNase treatment. This assay can be used to check whether mutations in the CP and RTP affect virion assembly. So this is an example of one mutant from Stewart's lab that has been tested in this way. This mutant contains 2 point mutations, resulting in a leucine to proline and a histidine to proline substitution. Although the viral genome is produced, we can see clearly in an RNase protection assay that virions are unstable or incomplete, indicating that this mutation disrupts capsid formation or stability. RNase protection and other classical approaches to studying Poleroviruses have connected many mutations in the CP and RTD to defects in virion assembly, movement in plants, and aphid transmission. Yet we're still missing something. How is it that these small changes in coprotein sequence lead to a change in function? And the missing link here is protein structure. These mutations can result in small or large perturbations in coprotein structure, which in turn may impact virion assembly or interactions with plant and aphid proteins. These changes are especially important for Poleroviruses, which move systemically in plants and aphids as virions. However, as I mentioned before, there are no publicly available crystal structures for any Polerovirus virion or structural protein. Without knowing what these proteins look like, how can we link these changes in sequence to disruptions in the viral life cycle? To try to approach this problem from another angle, we applied Protein Interaction Reporter, or PIR technology. PIR technology utilizes a mass spec compatible crosslinker developed by our collaborator, Jim Bruce, at the University of Washington in Seattle. This is a schematic of the PIR crosslinker. At the bottom are 2 reactive groups which react with solvent-exposed lysines on the surface of proteins. Applying this crosslinker to a mixture of protein complexes allows us to freeze protein-protein interactions. After crosslinking, we break the proteins into smaller pieces by digesting with trypsin. A biotin tag on the crosslinker allows us to enrich for cross-linked peptides before we run the samples through the mass spec. Next, a mass-coded reporter at the center of the crosslinker allows us to pinpoint cross-linked peptides in the mass spec, targeting them for further analysis. Finally, the mass spec cleavable bonds connecting the reactive groups to the rest of the crosslinker enable isolation and fragmentation of the peptides in each cross-linked pair. The mass spec cleavable bonds are what make PIR revolutionary, as they're what allow us to easily determine which peptides are cross-linked to one another. Application of the PIR crosslinker to protein complexes yields several types of crosslinks. Some, like these ones in pink, occur between 2 interacting proteins. The crosslinker can also react with 2 nearby reactive sites on the same protein, as shown by the yellow arrows. Finally, we see some dead-end crosslinks, like these ones in black, between reactive sites and water. These crosslinks are actually still useful because they tell us which regions of the protein are solvent exposed. Because PIR technology identifies not only which proteins interact with one another, but also which residues were cross-linked, it gives us information that we can use for structural modeling. This is great because computational modeling without any other information often results in hundreds or thousands of possible models, as shown by the myriad of ways we can imagine putting this green protein and blue protein together. However, finding a crosslink, for example, between the red dots on these proteins, tells us that these red dots have to be near enough for a crosslinker to connect them. Using the length of the crosslinker arms as a distance constraint, we can instantly eliminate many of the possible models. This brings me to the research portion of my talk, which I'm going to divide into two sections based on the type of crosslinks that we found. In the first section, I'll talk about some work in structural biology of Poleroviruses using PIR-Guided modeling. In the second part of the talk, I'll detail how we used PIR to explore host-virus interactions. I also want to remind you that my experiments are going to alternate between the use of our two model Poleroviruses, potato leaf roll virus and turnip yellows virus. Okay, so let's talk about structural biology. In the absence of a crystal structure, we can use PIR to make guided predictions using computational models. And in fact, Michelle Heck, Juan Chavez, and Stacy de Blasio have already used PIR cross-linking data to generate computational models of the PLRV coprotein trimer. And my goal in this first section of my talk is to extend these techniques to TUIV. Even though the coprotein sequence is remarkably well conserved across Poleroviruses, these viruses, PLRV and TUIV, have different hosts and can cause different symptoms. So we didn't know whether the interaction topologies would be conserved. To apply PIR to study Poleroviruses, we first do a protein extraction from infected tissue. Then we do a crude virion purification, which enriches for virions while retaining some host virus interactions. Applying the PIR crosslinker to this sample lends data about interactions between viral proteins, as well as interactions between viral and host proteins. Importantly, the aphid transmissibility of every crude virion purification is tested prior to crosslinking to ensure that the samples contain intact, biologically active virions. So here's what we found when we applied this workflow to TUIV. This is a schematic of the TUIV coprotein sequence shown in blue with the optional read-through domain shown as an orange dotted arrow. A lysine at position 150 in the coprotein was found cross-linked to another lysine at position 179. An important limitation of PIR is that we can't tell whether these cross-linked lysines were on the same coprotein monomer or on adjacent monomers, which limits our ability to use cross-links like this one for modeling, although they're still useful. Remarkably, this exact crosslink was also found in PLRV. We can be certain that these crosslinks are truly homologous to one another because not only are the amino acid positions similar, but also the peptides found crosslinked were nearly identical, differing by only a few amino acids. We also found a crosslink between two identical lysines found at position 179. In this case, we know that this crosslink had to occur between two adjacent CP monomers because this peptide only occurs once in each molecule. We call this kind of crosslink an unambiguous homodimer, and this is the most useful kind of crosslink for modeling. This crosslink was also perfectly conserved in PLRV. So now that we have some cross-linking data, how do we use it for modeling? First, I used a tool called iTASR to predict the structure of a single coprotein monomer by homology modeling. You can see the structure here. It consists of an S or shell domain, which forms the surface of the capsid, and an R domain, which interacts with RNA inside the capsid. Next, I model the multimer. It's not computationally feasible to model the entire virion, so instead we choose a subset. In this case, the coprotein trimer, which is the basic unit of the capsid. The classical trimer makes up one face of the virion. I use SimDoc to assemble the monomer into plausible trimers in as many different ways as it can, while keeping my cross-linked residues within a certain linear distance of each other, based on the length of the PIR cross-linking arm. Without the distance constraints, this analysis produces hundreds of models, but our cross-linking data narrows it down to only 77. Finally, I fed these 77 models into a script which calculates the shortest solvent-accessible distance between the cross-linked residues. This is important because linear distances often pass through other parts of the protein, which our cross-linker can't do. You can think of this as the difference between going from here to Australia by tunneling through the planet versus taking an airplane over the planet's surface. After all of these calculations, I ended up with 2 trimer models that fit our distance constraints. So those 2 models are shown here with the cross-linking sites used for distance constraints, shown space-filled in different colors. When we compare these to the most recent PLRV trimer model from Stacy de Blasio's paper, we can see that model A is more similar in shape, but still isn't an exact copy. Part of the reason we obtained 2 rather different models for the trimer is that there are many different ways that monomers contact each other in virions. For example, these pink points are a 3-fold axis of symmetry at the center of the classical trimer, whereas the yellow and green contacts occur at the vertices of the virion. Our model changes depending on where in the larger 3D structure we think the crosslinks occurred. Model A, as well as Stacy's model, fit best at the 6-fold axis of symmetry, as shown here. Model B, on the other hand, places the crosslinks at the 3-fold axis of symmetry, making this a true trimer model. So you might remember earlier I mentioned this LH mutant from Stewart's lab. This is the one that he tested by RNAse protection and found that it doesn't make stable virions. So if we highlight these mutated residues on model A of the TUYV trimer, we can make some predictions about why this might be. These 2 mutated residues are located near the 3-fold axis of symmetry in this model, which is likely a really important area for assembly and stability of the trimer. Additionally, the histidine at position 108 has a positively charged side chain and is near a negatively charged aspartate on the neighboring monomer. Mutating this histidine to proline, which is hydrophobic, would greatly disrupt the salt bridge and favorable interaction. This is just one example of how a structural model can be used to bridge the gap between sequence and function. So what's next? The high degree of conservation of structural crosslinks between TUYV and PLRV provides grounds for extension of these data to other Poleroviruses. This is important because it allows us to make structural predictions for Polerovirus species that don't yet have well-developed toolboxes, including newly emerging species. We also, of course, would like to be able to confirm our predictions by solving some structures. And in fact, we're doing this in collaboration with Josh Chappie, who's an expert in X-ray crystallography and cryo-EM here at Cornell. He's in the audience today. And actually, I did some early work trying to solve the structure of the PLRV virion by cryo-EM, but didn't have time to overcome some of the technical difficulties we had early on. Josh's lab is also trying to solve the crystal structure of the structural proteins. Carl Schiltz, a grad student in Josh's lab, is actually very close to solving the structure of the coat protein. And he is also working with Jenny Wilson, who's a grad student in our lab, to solve the structure of part of the read-through domain. And the results from this project have been really, really exciting. And if you want to hear more about them, I would encourage you to look out for their papers or to try to visit one of their seminars on campus. In the meantime, these PIR data are the only measurements we have of structural proteins for Poleroviruses. And we expect these data will continue to be important even as we get closer to solving the structures. So this paper is an example of a structure which was obtained primarily through cryo-EM, but utilized cross-linking data to resolve an area that was otherwise ambiguous. Before I move on, I want to mention one more thing. Although all of the virus-virus interactions I've talked about so far have been between structural proteins encoded by ORFs 3 and 5, PIR can also detect interactions with any other viral proteins that are present in our crude virion purifications. And in fact, my data set included 2 crosslinks between the TUYV coat protein and a lysine located in the peptidase domain of the viral P1 polyprotein. This interaction has never been shown before and is interesting because of a very old mystery in the Polerovirus field. This is an image of a Western blot using an antibody that recognizes structural proteins. The far left lane contains crude protein extracted from PLRV-infected plants. You can see that there's a clear band visible for the coat protein at 23 kilodaltons and for the full-length read-through protein at about 80 kilodaltons. There's also a faint signal between 50 and 70 kilodaltons, which is hard to see here. However, if we take this same plant tissue and use it for a virion purification, we see a very different story. The coat protein band remains as expected, but the full-length read-through becomes difficult or impossible to detect. Meanwhile, the faint bands we saw earlier resolve into a handful of clear bands between 50 and 70 kilodaltons. These bands are believed to correspond to truncated forms of the read-through protein. For many years, researchers weren't certain whether these truncations were an artifact of the purification protocol or whether they had some biological function. However, this paper published in 2014 showed for the first time that the truncated RTP did have a function. Authors found that both the full-length and truncated forms of the RTP were absolutely required for efficient systemic movement in plants. They hypothesized that only the truncated RTP is incorporated into virions, accounting for its enrichment in purified preparations, but that unincorporated full-length RTP interacts with other host proteins to facilitate movement. However, the authors were not able to determine how RTP truncation occurred. My finding of a cross-link between the CP or RTP and a lysine in the peptidase domain of another viral protein suggests that Poleroviruses may perform this truncation using their own viral protease. This is supported by an analysis of the locations of the putative P1 cleavage sites in the RTP of Poleroviruses, as well as others in the family Luteoviridae. In the coat protein and N-terminal half of the read-through domain, there are very few putative P1 cleavage sites. However, we see a massive increase in the frequency of these sites in the C-terminal half of the RTD. Cleavage at these sites would result in truncated RTPs of 50 to 70 kilodaltons, which corresponds exactly with the sizes that we see on a Western. So in this portion of the talk, we found that these structural crosslinks are conserved between PLRV and TUIV. We found a cross-link between a viral protease domain and the coat protein, which led us to postulate a possible mechanism for RTP truncation. And we expect these data to be useful in the future as we integrate them with crystal structures. Next, I want to talk a little bit about host-virus interactions. As I mentioned previously, PIR can also be used to probe host-virus interactions, as these are preserved during our crude virion purification. So when I first joined the HEC lab, Stacy de Blasio was working on this project. She found interactions between PLRV coat protein or read-through protein and 32 different plant proteins. This one is my personal favorite. It's a protein called BIP, which is a heat shock protein. And when Stacy modeled the interaction between BIP, which is shown in blue, and the PLRV coat protein, shown in green, she found that the coat protein actually fits perfectly into BIP's substrate binding pocket. Here you can see a web logo analyzing the conservation of the BIP binding site among the coat proteins of other viruses in this family. The size of the letter corresponds to the degree of conservation. Not only are the amino acids that are directly involved in binding highly conserved, they're also exactly the same motif that BIP is known to bind to in host proteins, indicating that this motif has evolved in viruses to mimic the host substrates of BIP. This is especially interesting because these residues are a short but highly conserved stretch of hydrophobic amino acids in what is otherwise a highly charged region of the coat protein. Seeing this, we wanted to explore this protein further. And one of my first projects here was to take this protein and a selection of others found interacting with PLRV and to try to determine what the purpose of this interaction was. Fortunately, there's already a very well-established system in place for gene silencing in Nicotiana benthamiana, our plant of choice. This system uses another virus, tobacco rattle virus, or TRV, to silence genes by virus-induced gene silencing. After inserting a fragment of a plant gene, like BIP, into one of the halves of the TRV genome, young plants are inoculated by agroinfiltration. As the virus replicates and moves systemically, homologous transcripts are silenced in new tissue. Since we'd never tried this system before, we chose a relatively simple approach to start. I inoculated plants with TRV containing a silencing construct and allowed them to grow for several weeks, as shown on the previous slide. I then inoculated several young leaves with PLRV by agroinfiltration, waited a few days, and sampled the infiltrated area by taking leaf punches to check titer by ELISA. Because this assay samples locally infected tissue, we expect to be able to assess changes in virion assembly, virus replication, and host defense. So here's what we found when we used VIGS to knock down BIP. This is a graph of PLRV titer in the control plants, which are infected with a version of TRV that shouldn't be silencing anything, and the plants where BIP expression has been knocked down. You can see that there isn't much of an effect at 3 days post-infiltration, or DPI. However, just 2 days later, PLRV titer has fallen in the control plants, but risen in the BIP silenced plants. I tested 11 different host interactors in this way, and found that knocking down 8 of them affected titer during early or late stages of local infection. In later PIR experiments, I also found 2 cross-links that we had not seen before, between PLRV coprotein or read-through protein and a plant Argonaut-1 or AGO1 protein. Although Stacy had previous evidence that AGO1 co-immunoprecipitates with PLRV, this was the first evidence we had of a direct interaction. Before I go into the follow-up experiments, I want to take a minute to talk about the role of AGO1 in antiviral defense. Plants' primary defense against viruses is RNA silencing. During replication of Poleroviruses and most other plant viruses, a double-stranded RNA version of the viral genome is temporarily produced. This dsRNA can be recognized by plant dicer proteins, which chop it into pieces. The resulting fragments can be small primary interfering RNAs, or siRNAs, 22 to 24 nucleotides long. These siRNAs are loaded into Argonaut proteins, including AGO1, forming the RNA-induced silencing complex, or RISC. This complex targets homologous transcripts for silencing by cleavage or translational inhibition. To combat this defense, most plant viruses encode a silencing suppressor. If you remember from the very beginning of my talk, the silencing suppressor protein for Poleroviruses is called P0. P0 is actually believed to function by preventing assembly of the RISC and promoting degradation of AGO1 protein. This does not prevent generation of primary siRNAs by dicer, but it does keep AGO1 from using these siRNAs to target other viral transcripts. Based on this information, we can hypothesize that knocking down AGO1 will increase PLRV titer. To test this, I came back to my VIGS approach. However, this time I inoculated young plants with TRV and PLRV at the same time. After 2 to 3 weeks, when both viruses had moved systemically, I took leaf punches from young leaves and assessed titer, again by ELISA. Because this approach assays systemically infected tissue, it surveys a larger portion of the virus life cycle. Here I'm showing the effect of silencing AGO1 on PLRV titer at 2 different time points. Each dot represents 1 leaf punch. Pink dots are from the control plants infected with TRV that shouldn't be silencing anything, whereas blue dots are from plants infected with TRV targeting AGO1. While silencing AGO1 did greatly increase PLRV titer in some samples, you can see that there was a huge amount of variation, even between samples taken from the same leaf. Because of the enormous variability between samples, there was not a statistically significant difference between the treatments. So after seeing these data, I wanted to see if I could alter my sampling method to reduce variability. So I reran the previous experiment, but this time I took 3 punches from the left half of the leaf, which I've labeled as 1, 2, and 3. This is a box and whisker plot to show variability, and here the control plants are in blue, and the AGO1 silenced plants are in pink. You can see that variability is lower in some areas of the leaf than others. Next, I excised the midrib of the same leaf as one sample and took the entire remaining right half of the leaf as another. You can see that the titer in both of these samples was pretty variable. Finally, I took the petiole of the leaf. PLRV titer was by far least variable in these samples, and a clear difference could be seen between the treatments, even though I'd sampled approximately the same number of plants as before. So with this in mind, I set out to do this experiment one more time. This time I took petiole samples, and I also tripled the number of plants in each treatment because I really was tired of doing this experiment. So here you can see that silencing AGO1 significantly increased PLRV titer relative to the control plants. You can also see that PLRV titer was significantly more variable in the AGO1 silenced plants than in the controls. You might remember that we saw this in the very first set of experiments I did too. I also checked TRV titer. You can see that silencing AGO1 also clearly increased TRV titer, which makes sense given that AGO1 has a role in general antiviral defense. However, unlike PLRV, TRV titer was similarly variable in the control and AGO1 silenced plants. This suggests to me that there's something unique about the relationship between PLRV and AGO1 beyond AGO1's role in broad spectrum virus defense. Although I was incredibly happy to see some low p-values after all of that optimization, I've actually saved what I think is the most interesting result for last. It's a little bit hard to see in this photo, but infection with TRV AGO1 alone, that is without PLRV present, causes some stunting and morphological defects. This isn't really surprising because AGO1 is known to have a role in transcriptional regulation during development. That said, these plants are still very much alive and will even eventually flower. PLRV infection, on the other hand, causes very mild symptoms, even when our control version of TRV is present. However, if you combine TRV AGO1 and PLRV infection, the plant withers away and eventually dies. To understand why this might occur, we need to think about all the factors influencing AGO1 levels in these plants. AGO1 protein is made by translation of AGO1 mRNA. When PLRV is present, it produces P0, which promotes degradation of AGO1 protein to facilitate virus replication. TRV AGO1 also works to knock down AGO1, but it does so at the transcript level. From this information, we can hypothesize that TRV AGO1 and PLRV individually knock AGO1 down, but not out. However, when both are combined, AGO1 drops to lethal levels. This is supported by work from other labs showing that AGO1 null mutants in Arabidopsis are lethal. All of this suggests that Poleroviruses must tightly control P0 activity. Knocking down AGO1 increases virus titer, but if you knock it down too much, the host dies. This brings me back around to the crosslink between PLRV coat protein and AGO1. What if the purpose of this interaction is to protect AGO1 from P0? This would be an elegant way to maintain AGO1 homeostasis. As virus titer and likely the production of P0 increase, more CP is also produced to counteract P0's effect. This hypothesis has mixed support from qPCR data. The expression of AGO1 is almost identical in control plants, regardless of whether or not the plants are co-infected with PLRV, meaning that PLRV infection alone doesn't significantly disrupt AGO1 homeostasis. However, PLRV infection has a noticeable effect in plants infected with TRV targeting AGO1. In fact, PLRV infection in these plants increases AGO1 expression, likely in an effort by the plant to compensate for P0's effect at the protein level, although it's important to note that this increased expression may not result in increased levels of AGO1 protein. So this seems to contradict our hypothesis. The presence of the coat protein should shield AGO1 from P0, especially because we know from the previous data that the titer is very high in these plants. But we already know that the coat protein can't be 100% effective at preventing AGO1 degradation, or there would be no point to having P0 at all. So maybe the plants are more sensitive to even the reduced action of P0 when AGO1 is already knocked down at the transcript level. I'm still working on testing this hypothesis, but I think there are interesting implications for the data I already have, regardless of how the next experiments turn out. Knocking down AGO1 affects PLRV titer in a much more complex way than TRV titer, suggesting that there's something unique about the AGO1 Polerovirus relationship. This is also supported by the crosslink we found between the coat protein and AGO1. We definitely have evidence that the strength of P0 is a balancing act between suppressing host defense and killing the host. And AGO1 homeostasis is not affected by PLRV infection alone. This led us to develop a hypothesis about the function of the CP-AGO1 interaction. So in this second half of the talk, we've assessed the effect of knocking down some host interactors during PLRV infection and developed a method to test these interactions using virus-induced gene silencing. We also found a direct interaction between AGO1 and PLRV coprotein or read-through protein. And we've seen evidence that Poleroviruses must modulate P0 activity to prevent host death. Okay, so I know I just gave you 6 years worth of data in 30 minutes. So I want to wrap things up by reiterating what I think are the main findings of this research. In the first part of the talk, we found that the interaction topologies of Polerovirus coproteins are conserved, which sounds relatively trivial, but is actually really important because if the coprotein interactions don't change between Polerovirus species with different hosts, this means that these species-specific differences are not encoded in the coprotein topologies. Since Poleroviruses move as virions in plants, we can hypothesize that host specificity must then be encoded in the read-through domain by the process of elimination. We've also demonstrated for the first time that PIR successfully identifies direct, functionally relevant interactions between Poleroviruses and their hosts. This is also not trivial. We knew of very few direct host-virus interactions previously, and finding these interactions was a matter of luck or screening using yeast-2 hybrid or other high-throughput approaches, which are very time and resource intensive and don't always identify interactions that are functionally relevant. Not only have we found many new host interactions to explore, but we've also made a huge technological leap forward in the field. Finally, I didn't show all of the data for this, but in my VIGS experiments, I found that knocking out most of the host proteins that we identified as interacting with Poleroviruses by PIR actually increased Polerovirus titer. And this was a really surprising result because we were expecting to find at least an equal number of interactions that are important for virus replication or movement as for defense. There are a couple of ways that we can explain this. It's possible that plants really just throw everything that they have at viruses to try to prevent replication. And so most host virus interactions actually represent the plant's attempts to defend itself. Alternatively, we know that circulative viruses, that the circulative transmission mode of Poleroviruses requires prolonged aphid feeding for acquisition and transmission. While it's beneficial for viruses with other transmission modes to sacrifice their hosts in an effort to produce as many virions as possible, Poleroviruses must keep their host alive and attractive to aphids. Maybe the reason we see an increase in titer when we knock out most host interaction partners is because the function of these interactions is actually to limit virus replication. To wrap things up real quick, I want to talk briefly about some potential future applications and impacts for this research. I'm really interested to see how these data could be applied to other non-model Polerovirus species, including some of the newer emerging viruses. These types of data could also be useful for the development of interdiction molecules, which are molecules specifically designed to interfere with the biological process. Angela Cruz, a grad student in our lab, is actually working on the development of interdiction molecules to prevent transmission of the bacteria that causes citrus greening disease. So if you'd like to learn more about that, then you can look for her papers and seminars. I also think that this technology has some potentially interesting applications for gene editing, and particularly in the development of resistant cultivars. So this is a paper from 2011 detailing the development of potato cultivars resistant to 3 strains of potato virus Y, which is a potevirus that's one of the most important potato pathogens today. Like Poleroviruses, PVY has a single-stranded RNA genome and encodes a viral genome leaked protein, or VPG, which binds to the 5' end of the viral genome. VPG then binds to a host elongation factor, in this case EIF4E, to recruit a host ribosomal complex to initiate production of viral proteins. Previous screens had identified EIF4E alleles in other plant species which were fully functional but contained small polymorphisms that prevented binding to VPG, conferring resistance to poteviruses. In this study, the authors used targeted mutagenesis to transfer those polymorphisms into potato. The resulting transgenic lines are numbered here on this graph, with the wild type and other control lines on the far right. You can see that all of the transgenic lines were almost completely resistant to all 3 tested strains of PVY, whereas the controls were highly susceptible. I believe that PIR-guided modeling has the potential to achieve the same end result for Poleroviruses, but in a much shorter time frame. We have already established that PIR successfully identifies functionally relevant host-virus interactions, which is the first step in developing a resistance strategy like this. We can also use PIR data to model the interaction between the host and viral proteins, predicting where the viral protein binds. If this binding site is located in an area that can be mutated without affecting the host protein's native function, we can perform gene editing to knock out the interaction without affecting the protein's native function. This is especially important because many of the host proteins we found interacting with PLRV and TUIV have important roles in metabolism and development and can't be knocked down or out without causing significant and sometimes lethal effects. And this is only the beginning. Now that we finally have the ability to more directly explore structural biology and protein-protein interactions, we can try to tackle some of the oldest and most important questions in luteovirid biology. How do Poleroviruses interact with their hosts? Can we interrupt virus replication or movement? What's the structure of the Polerovirus Virion and what governs its assembly? Can we design molecules to prevent assembly or disrupt host virus interactions? How do Poleroviruses cause symptoms and yield losses in host plants? Can we use this information to reduce the impact of these viruses? And finally, aphid transmission is largely a black box. Application of PIR to aphid virus complexes could greatly help us to understand circulative transmission of these and other viruses. So overall, I think these data and this technology are really exciting and give us the ability to leap years ahead in our understanding of Polerovirus biology. And I hope I've convinced you of that too. Before I take questions, I'd like to take just like 1 minute to thank a couple of the people who have made all of this possible. So I've been fortunate to have many amazing mentors in my time here. My advisor, Michelle Heck, has been a constant source of support and inspiration. Anyone who has met her knows that her seemingly endless energy and capacity to generate new ideas is truly incredible. And from Cold Spring Harbor to many government shutdowns, we've been through a lot together. And I'm so grateful to have been able to take this journey with her. I also want to thank my committee, all of whom are amazing, and I'm so honored to have been able to have their feedback. Also, Stacy de Blasio laid the foundation for so much of this work and has been an incredible sounding board, friend, and teacher, and I could not have done any of this without her. And to my many other mentors who have helped me to become a better scientist, a better teacher, and a better communicator, thank you. I know everyone says this, but I truly have the best collaborators in the world, not only Jim Stewart and Josh, who I mentioned previously, but also Veronique Brault and Veronique Ziegler-Graf, who enabled all of the work with TUIV. My husband, Dave, has been my rock through even the hardest days in the lab. Not only has he been unwaveringly supportive and encouraging, but he has personally contributed to this project through many late nights helping me inoculate plants. I would also not be here today without my family, especially my sister Midori and my parents, pictured here in their adorable Christmas attire, who have been a constant source of strength, inspiration, and love. You guys are the best, and I'm so lucky to have you. Finally, I'm so grateful to have met so many brilliant, kind, and creative people here. And I especially want to thank Angela Cruz, who has had the misfortune of spending so much time with me that we've started taking on one another's mannerisms. She's picked me back up so many times after I've fallen, and I truly don't know what I would do without her. So thank you all of you for your support and for listening today. It means a lot to me. And at this point, I think we have time for a couple of questions. So I'll start by taking a question from Ithaca. Andy.
So I really like this AGO hypothesis about the coprotein kind of modulating degradation of AGO. And you showed in your model, or you showed your data that says if you have a VIGS knockdown plant that's inoculated with the virus, you see this like dead plant, and you're saying that that could be due to AGO going below the threshold for. So what do you think would happen if you overexpress the coprotein or try to play with that system?
Yeah, so Andy was talking about this AGO1 story and my hypothesis for the coprotein interaction protecting AGO1 from P0, and was wondering if maybe if we overexpress the coprotein or did something else to kind of mess with the system, if we could maybe provide some support for this hypothesis. And that's actually is something that I'm working on. I'm using something called a silencing suppression assay, which I just barely didn't get done in time for this talk. But basically, I'm trying to see if I can overexpress the coprotein and see a difference in how well AGO1 is degraded by P0. Josh?
So you showed this sort of proteolysis of your RTD, right?
Yes.
With the viral protein. Do you know how that maps to our structural data? Like, does it correspond to where we see structurally, or does it line up to the point where we have structure?
I don't remember off the top of my head. I have looked at that, and I can't remember what the results were, but that would be really interesting to check to see where those putative cleavage sites are. Yeah.
I mean, so the cleavage sites are in the C-terminus of the NR. of the RTD.
Yeah, but she showed a distribution, right? So and it drops off very, very, very strongly at a certain point. My question is, we have a structure of the N-terminal part of it, so does that map to where that drops off or not? And what we need for the structure, that's all.
Well, and I think it would also be interesting to see whether the putative cleavage sites that we do see in the NRTD are maybe protected in the structure. So maybe they could be present, but aren't being recognized by P1, which could explain why we never see cleavage there. So yeah, definitely. Are there any questions from Geneva or other remote audiences? No? I think we're about out of time, but if anyone has any other questions, feel free to come and grab me afterwards. So thank you guys.
This has been a production of Cornell University, on the web at cornell.edu.